Hello everyone! In this video, I'm going to talk about the synthesis of Cephanolide A, a natural product that belongs to a family of diterpenoids that have shown a broad range of bioactivity, including antiviral and antitumor properties as well as cytotoxic activity against human cancer cell lines. From a retrosynthesis standpoint, the authors aim to maximize the rapid generation of the structurally complex carbon framework of the molecule. To this end, identifying the strategic bonds that needed to be forged could simplify not only the preparation of this molecule, but also any of the other members of the family, just by changing the sequence of the late-state oxidations. An intramolecular inverse demand Diels-Alder cycloaddition was identified as a strategic disconnection for the efficient construction of this carbon framework, and from this intermediate, the authors identified two fragments, a pyron and an indanone, that could be linked with a two-carbon fragment by an iterative cross-coupling sequence. The synthesis commenced with the triflation of this commercially available indanone. Then, an sp2-sp3 Suzuki cross-coupling took place with this organoborane, which was generated in situ from hydrogenation of the vinyl reagent. The Suzuki coupling was accomplished with palladium diacetate as the catalyst in the presence of DeFos as the ligand and potassium fluoride as the base. The product was then subjected to another Suzuki coupling with pyron triflate. In this case, they employed Rufos as the ligand and potassium carbonate as the base. The key intramolecular Diels-Alder reaction was accomplished upon formation of the silyl-enol ether with two equivalents of trimethylsilyl triflate. The first equivalent for the formation of the enol ether and the second equivalent, presumably, as a Lewis acid to facilitate the cycloaddition that led to the endocyclohadduct. The next step was the functionalization of the bridging olefin group. To this end, several hydroborations and epoxidation reactions of the olefin group were attempted without success. Fortunately, a modified Mukaiyama hydration protocol proved effective. The Mukaiyama hydration allows the transformation of an olefin to the corresponding alcohol with Markovnikov selectivity. A plausible reaction mechanism suggests that the reaction proceeds through a cobalt peroxide adduct that leads to acylyl peroxide that is converted to the alcohol upon reduction. In this modified protocol, a nonaflate peroxide is generated instead. Then, DBU induces elimination and formation of the corresponding ketone. According to the authors, the retroselectivity of the hydrocoagulation likely results from a directing effect of the proximal oxygen lone pair of the lactone. Besides, the reaction had to be carried out at minus 78 degrees Celsius because the excess DBU at higher temperatures caused enolization of the resulting ketone and the carboxylation of the strained lactone. The subsequent olefination reaction was attempted with several olefination reagents. For instance, the Wittig olefination failed because phosphorus ylides proved to be too basic even at cryogenic temperatures and resulted in the opening of the lactone. Finally, they found an olefination protocol, which employs less basic olefination reagents and that gave the desired hexamethylene group. The Rayleigh oxidation resulted in the formation of diallylic alcohol, which was subsequently oxidized to the corresponding enone. Hydrogenation, followed by epimerization of the methyl-bearing stereocentre, gave the desired ketone in two steps. The ketone was then reduced with sodium borohydride to the corresponding alcohol. The alcohol was subjected to Suarez oxidation conditions, employing iodine and hypervalent iodine to forge the desired THF ring. After TMS cleavage, the tertiary alcohol was obtained. The alcohol was eliminated following the classical Barton-McConville deoxygenation reaction. Final oxygenation of the arene moiety using cyclopropane malonyl peroxide completed the synthesis of cephanolide A.